Now let us continue with part 3 of the study of eukaryotes. Going back to our overview of eukaryotes, we will focus on the internal structures of the cell. The internal structures of the cell are organelles, and they have been divided into membranous and non-membranous organelles. This part 3 will focus on the membranous organelles, which are the endoplasmic reticulum, the Golgi Apparatus, the lysosomes, the mitochondria, and the peroxisomes. Let us study the endoplasmic reticulum first. The endoplasmic reticulum, also known as ER, includes the smooth and the rough endoplasmic reticulum. The ER is a network of interconnected sacs known as cisternas that interconnect with the nuclear membrane, as seen over here. The smooth endoplasmic reticulum, also known as SCR, is shown here in yellow, and it is so called because it has no ribosomes on it, and therefore it has a smooth surface. The smooth ER is responsible for the transport of lipids. The rough endoplasmic reticulum is shown here in light blue, and it is so called because of the presence of ribosomes on its surface, giving it its rough appearance. The rough ER is responsible for the transport of proteins. The Golgi apparatus. This apparatus is a group of vesicles that has a cis phase and a trans phase to it. The Golgi processes, sorts, and packages lipids and proteins that come from the smooth ER and the rough ER. So, vesicles from both the smooth ER and rough ER transport their contents to the cis phase through the incoming transport vesicles, and after being processed, sorted, and packaged within the Golgi, the outgoing transport vesicles are formed and exit through the trans phase of the Golgi apparatus. Now, lysosomes. Lysosomes are circular type sacs that contain digestive enzymes, mainly acid hydrolases, that help with digestion at the cellular level. That is, they break down biomolecules such as nucleotides, proteins, amino acids, and these come usually from waste products and cell debris. Now, mitochondria. The mitochondria is an organelle made up of many folds containing an internal membrane and an external membrane. The internal membrane has creases that are called crista and an internal space that is called the matrix. Mitochondria are responsible for transformation of chemical energy into cellular energy in eukaryotes. The last membranous organelle we will study are the peroxisomes. Peroxisomes are sticky carbohydrate structures. They are responsible for the breakdown of very long fatty acids through the process of beta oxidation. This is the first step in the breakdown of fatty acids. Here is a peroxisome shown in an electron micrograph. And in this picture of fluorescent microscopy, the green dots represent hundreds of peroxisomes within the cell, which has this as its nucleus. So these peroxisomes correspond to these small green dots. So, as a recap, the smooth endoplasmic reticulum transports lipids and the rough endoplasmic reticulum transports proteins within the cell. The Golgi apparatus processes, sorts, and packages these lipids and proteins from the ER. The lysosomes aid with digestive functions, and the mitochondria is responsible for cellular respiration. Finally, the peroxisomes help with breakdown of fatty acids. This concludes the study of the membranous organelles of the eukaryotic cells.